just going to give you a little bit more detail about how you can help WormBase by contributing your data. So community curation is actually really important to WormBase, and that's for several reasons. First of all, as the authors and the people who have actually done the work in the lab, you are the subject matter experts. So you are the people who are really in the best position to know what is most critical about the data that you've generated in the lab. Secondly, the community curation pipelines that we have rely on structured entry forms. And that actually makes it a lot easier for us at WormBase to take data that's been published in papers that might exist as natural language and integrate that into a database such as WormBase. And lastly, and also very important, is that community curation actually really helps reduce the curation backlog that we have at WormBase. And so just to give you an idea of what that backlog is and what we're facing, over the years, this graph actually shows the increase in the number of C. elegans papers that have been published and are available in PubMed. And as you can see, it continues to increase. And this is partly for several reasons. Every few years a new technology comes along that is great for the community, but it also often can generate a lot of new data for us to handle. In addition, and equally important, is that individual papers in the literature increasingly have more and more data that we need to curate. So any help that we can get from the community is great and allows us to get C. elegans data into WormBase much faster. So I'm going to talk specifically about 3 different avenues by which you can help WormBase get your data in. The first, as Paul mentioned, is what we call author Firstpass, and I'll explain a bit more about what that is and how that works. We also have data type specific curation forms for data entry. I'll show you an example or 2 of those. And then lastly, I'll talk a little bit more about the micropublication pipeline. Okay, so firstly, author Firstpass. So this is actually a pipeline we've had in place at WormBase for almost 10 years. And this is where authors tell us what type of information and what kinds of entities are in your recently published papers. So our previous pipeline, we would send emails out on a weekly basis to corresponding authors and then ask you to fill in a form, list the gene names that are in your paper, the variations, or the data types such as interactions. And we had about a 40% response rate over the years, which is good, but we'd love to have it be better. But the point was, this was nearly a fully manual process for authors. So what we have started, actually, just the beginning of this month, is a new author Firstpass pipeline that we're really excited about because we think this will actually really lower the barrier for people to tell us about the data that's in their paper. So I'll just briefly explain how this works, and a lot of this relies on text mining and AI that we do in collaboration with the Texpresso Central text mining platform that was developed as part of WormBase at Caltech. So basically the way this works is that when your paper is published and we have the final version of the PDF at WormBase, we will process that to convert the PDF to text. And then what we do with our AI is we go through and we identify the different types of data in the paper. So for example, things like interactions or RNAi phenotypes. Using standardized nomenclature, we will extract entities that we find, things like gene names, variations, transgenes. We will then extract the corresponding author email, and then you will get an email in your inbox, so look for it, check your spam folder just in case, where we will actually then ask you to verify the information that our Texpresso-based pipelines have pulled out of the paper. So this is what the new author Firstpass form looks like. It was organized in a very similar way to the WormBase gene pages. So there are sections here that you can step through. starting with, say, an overview of genes and species, and each time you finish a section, you'll see a check mark to let you know you've completed that. But here I'm just showing you an example of what this looks like for genetic information. So for example, what we will show you is, given the text mining, these are the list of the alleles that we've identified as being in your paper. And we're actually using some threshold values for entities to decide which of these alleles or genes or species, etc., will present, because we're really looking for, say, the genes and the variations that are primarily the focus of the experiments in the paper. And so as an author, what you can do is you can just scroll through this list, and if you're happy with the list we've presented, you don't have to do anything. You can just say submit and move on to the next page, reagents. Or if there are things that you think we've identified, they might be in the paper, but they're not the main focus of the paper, you can remove them from the list. If we've missed things, and this is particularly important, then you can add them. And adding is also from lists of WormBase approved entities. So I also want to point out that this is why it's really important when you publish your papers that as much as possible you use standardized gene names and variation names that we have in WormBase. That makes it a lot easier for us to identify what's in the paper. So for data types, in many cases we are using our AI approaches at WormBase. So for those, you'll see a little icon that says TPC powered. So we will take the text and basically looking at the kind of words and the language that's in the paper, we can identify whether there are things like allele sequence changes or interactions. If so, what type. If it's correct, again, you don't have to do anything. You can leave it checked. But if it's incorrect, you can take the check off, or if we've missed it, you can add it. If you want, for different data types, you can also add critical experimental details. That helps the WormBase curators know what they need to hone in on for curation. Or in some cases where these exist, we've actually added link outs to structured data forms so that you can add additional information, but in a structured way, which again makes it a lot easier for us to get the data into WormBase. So, like I said, this is new. We just started sending emails out to authors at the beginning of this month, so we have a few more features we'd like to add. Of course, as new entities or data types come along, we want to make sure that we keep up and we have ways to extract those. We are also really interested in exploring how we can actually go from just saying there's a type of data here or this particular entity to actually retrieving these sentences in papers that might describe some of the data we curate, and in this example, might mention specific mutations, and then we can use some of our AI to say, well, this looks like this might be this phenotype, say dendrite development variant. Again, did we get it right? And if we did, as authors, you don't have to do anything. And then we're also working on sort of an author summary view page that will be embedded in WormBase. And this will allow you to go in, log in with your email, and see all the papers for which we've associated you as an author, and you can see whether or not it's been through our Firstpass pipeline, and if it hasn't, you can go ahead and do that, and that would be a huge help to us. Okay, so next I want to just mention a little bit about some of the specific data submission forms that we currently have at WormBase. So if you're on the WormBase homepage, and you go on the top right, there's a button there that says Submit Data, and this is where you can access some of these forms. So basically we have two different areas on this page. So there's one we call Community Curation that has things for phenotype data submission, allele sequence data submission like I just showed you. I'll talk a little bit more about micropublications after this. And then gene descriptions, which we're probably actually going to be phasing out because we now have a great way to do that through an automated pipeline. We also have other types of structured entry forms for specific types of data. A few things I'll point out here, for example, are updating your contact information which is really helpful for us at keeping track of our users, as well as things like gene sequence links and requests for new gene names, which again is really important to do before you publish. That helps make it much easier for us to identify things. So for example, the phenotype form, you can submit your phenotype information to WormBase using this online form. It will step you through what kind of information we're looking for, and then there's an optional section at the bottom of the page, again, to add a lot more detail. So a phenotype submission pipeline is another pipeline we have where we actually email you. We will email the authors every 3 months, and we'll alert you to old and new papers that we have in WormBase that have not been curated for phenotype yet, but for which we ask your help in getting that information. And then also, like I said, there's additionally a form for submitting allele sequence information. And we are very grateful for any of the community curation that we get. And so we list on our site the top community curators, and this just gives us a way to recognize how much we appreciate the submissions from our users. So lastly, and very quickly, I want to talk a little bit more about micropublications that you've heard about from Paul's talk. So micropublications can also, the website for micropublication can also be accessed from the WormBase homepage. This is a project that got started at WormBase, but is expanding considerably to other organisms and to other scientific fields. So this is the micropublication biology homepage. So if you're interested in more of the details about micropublications, please visit this page. But basically, here's what they are. So these are meant to really be capturing standalone experimental results. The key thing about micropublications is that they are peer-reviewed. They're open access. You'll get a registered DOI for them, so they are citable on your CVs. They aim to have a really fast publishing turnaround. They will, if they're not already, soon be indexed in Europe PMC, and there is no submission charge. So for some of the data types that we take as micropublications, again, there are structured submission forms, which just makes it that much easier to get the data into WormBase rapidly. And this is an example of a micropublication that led to an expression pattern in WormBase. So you can see here, this is now fully incorporated into WormBase, and as a reference, there's a reference to the micropublication. So we strongly encourage people to think about submitting a micropublication. There's more information under Submit an Article on the website, and there are some FAQs there. And if you want more information, you can certainly also email that. I really appreciate any contributions to WormBase through our author Firstpass, the data type specific forms, or micropublication. And any questions at all, feel free to contact us.